Okay, very good morning, my dear friends. This is Dr. Padala Tirupati, Assistant Professor, Department of Botany, SRR Government Arts and Science College, Autonomous Karimnagar. In this regard, we would like to invite today's our eminent resource person, Dr. A.Shailaja Madam Garu, Assistant Professor, Department of Botany, Government Degree College, Autonomous Siddipet. Madam is going to explain about the most important area in plant sciences, that is plant anatomy. Previously, Madam has clearly explained about the different types of tissues, simple tissue, complex tissue, and special type of tissues, and also meristematic tissues and the related theories. So, Madam, please welcome and share your screen and start your session, Madam. Please welcome. Madam, please share your screen. So, Madam is sharing screen. So, my dear friends, please wait. During the class, if you have any doubt, please ask without any hesitation. Madam is going to clarify your doubts. So, this is most important topic, my dear friends.
Good morning, sir. I am audible, sir.
Yes, Madam, you are audible.
Screen is visible, sir. PPT is.
Respond, my dear friends. Screen is visible. Pooja, Narmada, Roja, Rakesh. Screen కనబడుతుందా?
Yes, sir.
Shall we start, sir?
Yes, Madam. Please, you may proceed.
Okay. Good morning to everyone. Today's our topic is vascular tissue systems. In the last class, in the anatomy topics, we have completed that. In the last class, epidermal tissue system is completed. Now we are going to discuss the vascular tissue system. So the plant tissue systems, vascular tissue system. Already you know what is about the vascular system. So in the introduction part, the vascular tissue system consists of a variable number of vascular bundles. So already you know what are the vascular bundles. So we have discussed that which one is called xylem, which one is called phloem. So that are located in the steel. Actually, that vascular bundles are located in the steel. The steel is the central cylindrical portion of the axis, commonly surrounded by the pericycle and endodermis. See here in that in this vascular system, you know this is the xylem, this is the phloem, this is the cambium, vascular cambium. In the central part, that is pith. Pith is located in the center. Actually, these vascular bundles are located in the steel. So, in the wherever we have seen in leaf anatomy and root anatomy and stem anatomy, we have found that the vascular bundles, these are main parts of the tissue system. So each vascular bundle that these are commonly surrounded by the pericycle and endodermis. However, in the systems of many seed plants, well-defined endodermis and pericycle may not be seen. Okay. So in some plants, we are observed that pericycle and endodermis are surrounded by the vascular bundles. But a well-defined endodermis and pericycle may not be seen in the seed plants. Therefore, Isaiah that 1948, the scientist, he discarded the term steel and replaced it the term that by the term vascular cylinder. Okay, he given that the instead of steel, he given that the term vascular cylinder. Isau, ESAU. Isau in the 1948 he discarded the term steel and that is replaced it by the term vascular cylinder. So in that each vascular bundle consists of xylem and phloem tissues with or without cambium in between them. So that in the vascular bundle we are found that the xylem and phloem. So these two are in between that cambium is present or not. In roots, separate xylem and phloem strands are found on different day. That the xylem conducts the water and mineral salts and the phloem conducts the food materials. So this is the difference between the xylem and the phloem. The xylem conducts water and mineral salts. So that the phloem conducts the food material, supply the food materials. That is the xylem and the phloem. The arrangement of vascular bundles is variable in different plants and different parts of the same plant body. They may be arranged in the form of a circular ring. See here in this diagram, you know that these are arranged as in the form of a circular ring. As in the case of roots and in the dicot stem, they may be found scattered throughout the stem as in monocot stems. So in the monocot stems, we are observed that scattered arrangement. So, in this, that in certain plants, the vascular bundles remain scattered in the well-defined pith. Pith is also present in the center. Such bundles are called medullary bundles. So, those vascular bundles having the center part that filled with the pith, those are called medullary bundles. Example, Mirabilis and Boerhaavia and Bougainvillea, Acheranthus, Amaranthus. These are the example for the medullary bundles. So, those bundles having the center part that filled with the pith, those bundles are called medullary bundles. Example for this, please note down. I am asking after the class these questions. Mirabilis, Boerhaavia, Bougainvillea, Acheranthus, Amaranthus. Okay. So vascular bundles may also occur in the cortex of the some stems. Such vascular bundles are known as cortical bundles. So in some plants, we are observed that these bundles may also occur in the cortex region. So that in the cortex region, those are present in the cortex. Such vascular bundles are known as cortical bundles. Example, Casuarina and Nyctanthus. Please note down. This is also a very important point. Nyctanthus and Casuarina. Casuarina, Nyctanthus having the cortical bundles. That vascular bundles are present in the cortical region. Okay. So what is the vascular bundle constituents? So this a vascular bundle of a stem consists of three major components: primary xylem, primary phloem, and cambium. See here. So in this diagram, you observe that what is that primary xylem and secondary xylem. See here. This is the cork cambium, epidermis. This is the cortex and bark, cork, primary phloem, secondary phloem. Those parts, sir, you have to see in the PPT. Okay. So that which one is called primary xylem? Xylem, that the xylem present in the primary plant body is called primary xylem. So in the primary plant body, we have to see that the primary plant body having the xylem, that is called primary xylem. It is differentiated from the procambium of the apical meristem. See here, this is the primary xylem. Okay, so it is present in the primary plant body. The first formed xylem elements constitute the protoxylem. Protoxylem is a complex tissue and consists of tracheids, vessels, and parenchyma cells. these what is the components and constituents of xylem, which are called xylem elements and which are called phloem elements. These all classes are completed. Okay, so that in the protoxylem, a complex tissue, and these consists of tracheids, vessels, and parenchyma cells. That the protoxylem, tracheids, and vessels show annular, spiral, and scalariform thickenings. Okay, these already we are discussed in the last classes. Okay, in many monocot stems, that non-functioning stretched protoxylem elements are collapsed and are then occupied by open canals, which are called protoxylem lacuna. Okay, for example, maize. In the maize plants, we observed that the protoxylem lacuna. So these are the special arrangement we are observed in the maize plant. The primary xylem elements which develop a later stage of growth are called metaxylem. Okay, so which one the plant body having the primary portion? So in the primary plant body having the xylem, that is called protoxylem. Okay, so after the development, after the latter stages, after the latter stage of growth, the xylem elements are developed in the stem or root, that is called metaxylem. It consists of reticulate and pitted vessels and some tracheids. So the vessels of metaxylem have bigger and wider cavities. Okay, so which are discussed that the elements of metaxylem and protoxylem. Depending upon the position of protoxylem in relation to metaxylem, the primary xylem of three types: exarch, endarch, and mesarch. These terms also we discussed in the last classes, but uh, however, I explained again. Which one is called exarch? Okay, the protoxylem lies towards the periphery of the stele and the metaxylem towards the center. Okay, so exarch means protoxylem lies towards the periphery of the stele, so and the metaxylem towards the center. The development of xylem is called centripetal. Okay, example for this: roots and seed plants, roots of seed plants. Exarch means protoxylem is arranged at the periphery and the metaxylem is the center. Okay, after the developing of the stem, that after developing that which xylem is present in the stem, that is the center part, but the protoxylem is arranged at periphery. Those are, that is we are called exarch, means outside anamata. Okay, outside it is present, protoxylem is present at outside of the stele, that is called exarch. So next, coming to the endarch, protoxylem lies towards the center and the metaxylem at the periphery of the stele. It's complete opposite. Exarch means outside, protoxylem annam kada. Same like that, endarch means that center, that protoxylem is present at the center, and metaxylem is development of xylem is centrifugal. This development is called centrifugal. Stems of seed plants. Exarch example, roots of seed plants. Endarch example, stems of seed plants. Please note down these, those are important. And next, coming to the mesarch. So what is the mesarch? The protoxylem is surrounded by metaxylem on both the sides. See here, very simple. Exarch means outside, protoxylem is present outside, and endarch means protoxylem is present at the center. Example for that, endarch stems of seed plants. And come to the mesarch, the protoxylem is surrounded by metaxylem on both the sides. The differentiation of xylem is both centripetal and centrifugal. Example for this mesarch: rhizomes and rachis of certain ferns and cycas plant and ferns. Okay, certain ferns. సో ఫెర్న్ మొక్కల్లోనూ మరియు సైకస్ మొక్కలో కనుక చూస్తే మనం ఈ మిజార్క్ కండిషన్ ని అబ్జర్వ్ చేయొచ్చు. మిజార్క్ మీన్స్ ఏంటంటే protoxylem ki both the sides arranged that metaxylem. Okay, that is called mesarch. My voice is clear? Please.
Yes, ma'am.
Yes, madam.
Okay.
You may proceed.
Okay, okay, sir. Thank you. So next come to the primary phloem. So come to the primary phloem. The phloem present in a vascular bundle is known as the primary phloem. So the phloem present is in a vascular bundle that is known as primary phloem. It is found away from the center of the axis, that is towards the periphery. So primary phloem consists of a sieve tubes, companion cells, and phloem parenchyma, and some fibers also are present in the phloem. So in monocot stems, that parenchyma is absent. Please note down this point. It's very, very important. In monocot stems, the parenchyma is absent. Phloem parenchyma is absent in monocot stems. Okay. So after the completion of the class, I am asking these this type of questions. Okay. Some important questions. Please note down. The first cells of the phloem to mature are known as protophlloem, and it consists of narrow seed tubes and it is found towards the periphery of the stele. But companion cells are rare or altogether absent. The inner portion of the phloem consists of a bigger seed tubes and is called metaphloem. So in the protophlloem, that narrow sieve tubes are present, but after the development, that in the we are observed that the metaphloem, that bigger sieve tubes are present in the structure. Okay, phloem structure. Metaphloem consists of sieve tubes, companion cells, phloem, parenchyma, and fibers. But in the monocot stems, parenchyma is absent. Please remember that point. Next come to the vascular cambium. In the dicot stems, a thin strip of promeristem is present in between xylem and phloem. See here. So you observe that vascular cambium. Okay. So this one. This one is vascular cambium. It's clear? Okay. This is the vascular cambium. Okay. So this vascular cambium is present in between the xylem and phloem. This meristem is called intrafascicular cambium or vascular cambium. We are called intrafascicular cambium or vascular cambium. So such a meristem is absent in monocot stems and roots of dicots and monocots. See here, please remember this, it's very, very important. Vascular cambium or intrafascicular cambium. This meristem is absent in monocot stems and roots of dicots and monocots. So vascular bundles with cambium are described as open, and those without cambium are called closed. So which one having the vascular cambium? Those are called open type of vascular bundles, and without cambium, those are called closed type of vascular bundles. Okay. So I'll show you. See here. This is types of vascular bundles, and conjoint, collateral, and open. Okay. So in which vascular bundles that cambium is there, yellow color cambium is there. So that the type of vascular bundles are open type of vascular bundles. Okay. So in some, see here, C1 is there, that is closed type of okay, C and D, conjoint, collateral, and closed type of. So which one not having the vascular cambium, those are called closed type of vascular bundles. Okay. PPT is visible? It clear, Nana?
Yes, ma'am.
Okay. So these the vascular cambium is a lateral meristem occurring parallel to the long axis of the organ. The cambium cells may be uniseriate or multiseriate. The cells are living with dense cytoplasm and nucleus. Vascular cambium produces secondary vascular tissues during the secondary growth. So the vascular cambium is made up of two basic kinds of initials. They are ray initials and fusiform initials. See here, these are the I'll discuss after these two terms that which one is called ray initials, which one is called fusiform initials. So the vascular cambium is made up of two basic kinds of initials. First one ray initials and second one fusiform initials. So which one are called ray initials? It gives rise to parenchymatous medullary rays. So after the developing, it gives rise to parenchymatous medullary rays, that is called ray initials. So which one are called fusiform initials gives rise to secondary xylem and secondary phloem. After the secondary growth, we are observed that after that development of secondary growth, we are observed that secondary xylem and secondary phloem. So those are coming from the fusiform initials. Okay. So fusiform initials. This is the difference between the ray initials and the fusiform initials. Those only the parenchymatous medullary rays are produced. So that from the ray initials, and which one gives rise to the secondary xylem and secondary phloem? Those are called fusiform initials. This is also important point, and how that secondary xylem and secondary phloem are formed and from the fusiform initials. Please remember this. It is also very important. And next, coming to the type of vascular bundles. So according to the relative positions of the xylem and phloem, the vascular bundles are classified various types. Okay. First one is radial. See here. Radial, I1 is radial. See here, this is one, this one, radial means the xylem and phloem are arranged on different alternating with each other. See here, this is the xylem, this is the phloem. Okay, so the xylem and phloem are arranged on different radii alternating with each other. This arrangement is the most primitive one. Example, roots of seed plants. Radial vascular bundles are present in the roots of seed plants. Okay, please remember this, radial. And conjoint, which one are called conjoint? See here, this is the B1. B1 is the conjoint. Conjoint means xylem and phloem combine to form a bundle and are present on the same radius. Conjoint bundles are again of two types. See here, this is the conjoint means this is the phloem, this is the cambium, this is the xylem. Okay, so those are arranged on same radius, conjoint, and pakkane untayi anamata. So same radius, but they are arranged from here, combined to form a bundle and are present on the same radius. These are again of two types. Which one? Collateral and bicollateral. See here. So D1 is the collateral. See here, collateral and closed, conjoint, collateral and closed. D1. See here, D1. So after that, F1 is conjoint, bicollateral. Okay, F1 is bicollateral. So which one is collateral? Phloem occurs only on the outer side of the xylem. See here, this is the phloem. Okay, blue color, which one it shows blue color, that is called phloem. So these pink colored are called xylem. Okay, so collateral means phloem occurs only on the outer side of the xylem. If cambium is present in between the xylem and phloem, that is called collateral and open type of. So in this D1, this is the closed, means because of that yellow color, this is the cambium. Okay, so cambium is absent, that's why we are called closed type of vascular bundles. Collateral, conjoint, collateral, closed type of vascular bundles. So in this in between the xylem and phloem, that cambium is present, open type of vascular bundles. Okay, so collateral example, this one example for this, stems of gymnosperms and dicots. Okay, conjoint, collateral and closed type of vascular bundles. Example, stems of gymnosperms and dicots also. So when cambium is absent, it is called collateral. Closed example, monocot stems. Open example is dicot. stems. Okay, so when the cambium is absent, that is called collateral closed, and example is closed vascular bundles, monocot stems. Don't confuse that monocot stems. In the monocot stems, vascular cambium is absent. Okay, so bicollateral means phloem occurs on both sides of the xylem. These bundles are usually open. Example, cucurbita stems. Okay, see here. F1 conjoint bicollateral and open. F1, see here, this one. Inner cambium, outer cambium. Yellow color cambium is there. This pink one is the xylem. Blue one is the phloem. See here, bicollateral means bi ante two sides anamata. Phloem occurs on both sides of the xylem. These bundles are usually open. Open means cambium undi kabatti open type of vascular bundles. Okay, this one F1 is called conjoint, bicollateral and open type of vascular bundles. That is bicollateral. So next concentric and amphicribal. See here, concentric means in the vascular bundle, one type of tissue completely surrounds the other. It may be amphivessel and amphicribal. See here, G and H. See here, this one. G and H. So amphicribal means the phloem lies in the center. See here, this is the phloem center. This one, blue color is the phloem. See here, what is the difference between G and I? Okay, here xylem is the center, here phloem is the center. Okay, the phloem lies in the center, it is surrounded by the xylem, this one, I1. So it is also called a leptocentric. Example, Dracena yucca. Dracena yucca. I example randi. I. Okay. So this is called a amphivessel. The phloem lies in the center. It's surrounded by the xylem. This is called a leptocentric. Example, Dracena yucca. Amphicribal means the xylem lies in the center and it is surrounded by the phloem. It is also called hadrocentric. See here, this is xylem is the center, xylem surrounded by the phloem. Phloem surrounded by the xylem, that is called G1. Okay, amphicribal. Example, fence, fern mokkalu. Okay, FERNS, fence. So that is the amphicribal. So in the concentric, these it may be amphivessel or amphicribal. So these two, G and I. The xylem is the center, it's surrounded by the phloem, and the phloem is the center, it's surrounded by the xylem. That is two or concentric vascular bundles. So what is the function of vascular system? Coming to the functions, conduction of water takes place through the xylem, and conduction of organic food substances takes place through the phloem. Already we know that water and minerals through the xylem, and food materials from the phloem. Secondary xylem provides mechanical strength to the plant body. So which secondary xylem is present after the development of secondary growth. So that secondary xylem provides the mechanical strength to the plant body. This is about the, see here. So in the plant vascular system, see here that vascular tissue, this is the phloem, this is the xylem. So what is the difference between the xylem and phloem? It is made up of two different kind of tissues. So in that vascular tissue system, it consists of a series of elements that come through tubes throughout the plant. So which one is phloem, which one is xylem, you know. Okay. Xylem consists of two types of cells known as tracheids and vessel elements. Tracheids and vessel elements from tube-shaped structures that provide pathways for water minerals to travel from the roots and the roots to the leaves. While tracheids are found in all vascular plants, vessels are found only in angiosperms. So see here, note this point, vessels are found in only in angiosperms. Coming to the phloem, so already you know this. While tracheid cells are non-living, sieve tube and companion cells of the phloem are living. Okay, so companion cells possess a nucleus and actively transport sugar into and out of sieve tubes. This is about the phloem. This is about the xylem. So, what are the differences? You observe that. Next, coming to the xylem and phloem, see here. This is the xylem vessel. This is the phloem vessel. What is the difference? See here. You observe that carries water and nutrients that enter the plant in the root up through the stem and out to all parts of the leaf. This is the xylem. See here. This is a one way only. Water and minerals through this vessel. It goes out from the roots to the leaf. No end walls between the cells. Thick wall.
మేడం శైలజ మేడం గారు, యువర్ వాయిస్ ఈజ్ నాట్ ఆడిబుల్.
Vascular bundles. So this is a phloem. This is a xylem. So in the xylem, in the phloem, so what is the difference between these tracheids and vessel elements? Arrangements are different. Come to the see here. This is the xylem vessel. This is the phloem vessel. See here. So xylem carries the water and the nutrients that enter the plant in the root up through the stem and out to all parts of the leaf. See, this is the one way only. In the xylem vessel, you observe that one way only that water and minerals goes through the vessel from the roots to the leaf. Thick walls stiffened with lignin. Okay, this is about the xylem vessel. Come to the phloem vessel. That is the difference. What is the difference between the xylem vessel and phloem vessel? Water and food are pass through the phloem vessel. So this is the two-way flow. Okay, two-way flow. Cells have walls with the perforations. Come to the phloem vessel. See here. This is in the xylem elements we are observed that perforation plates arranged like this. Okay, this is a vessel, this is a tracheid, and this is the fiber. These are the xylem elements. And come to the phloem elements. This is the sieve plate, this is the companion cell, this is the sieve element, this is the fiber. So these xylem fiber and phloem fiber are same structure. Come to the xylem tissue and come to the phloem tissue. So what are the differences from the xylem and phloem? See here, in the xylem we are observed that tracheids and vessels. In the phloem tissue we are observed that companion cells and sieve tube elements. So what is the main difference? So in the companion cells, parenchyma cells adjoin the sieve tube cells. They assist the sieve tube cells in the conduction of food. They are living cells with elongated nucleus. So see here, remember this. These are the living cells. Okay, the companion cells are the living cells. And come to the sieve tube elements. These are also elongated cells placed end to end to form a tube. Their end walls have pores in the which have the sieve plates. So the main function of the sieve tube is to carry the food, especially carbohydrates. Remember this. Okay. Come to the xylem tissue. So in this, the tracheids are there. These are the dead cells. Tracheids are dead cells with lumens when mature. Okay. So in the vessels are dead at functional maturity and are perforated. These are the vessels. These are the dead cells. Okay. So these are the main important topics of vascular bundles. Okay. So next I'm going to start fundamental tissue system. So please wait. Ground or fundamental tissue system. Am I audible? Sir, I continue the class. Time is there, sir. Please, Tripat, sir.
మీరు ప్రొసీడ్ కండి మళ్ళీ కంటిన్యూ
Okay, okay. Sir, ఒక half an hour, sir.
చేయండి మేడం.